Welcome back to part 4 of the lecture concerning the biosynthesis of amino acids. As I have been fond of mentioning, glutamine and glutamate are carriers and donors of nitrogen for biosynthesis in our organisms. At this point, I will also add that in bacteria and plants, glutamate synthase enzyme we have not encountered before can combine glutamine and alpha ketoglutarate to form two glutamates. Although the synthetic pathways for nitrogen incorporation into biomolecules are similar across organisms, this is an example of how bacteria and plants often have biochemical tools that animals do not have. Remember the transamination processes covered in part 2 and the transfer of amide nitrogen from glutamine to ammonia in part 3. These are also important in molecular synthesis pathways that we will discuss in this part. In addition, the transfer of one-carbon groups and amido transferases need to be covered briefly before we move forward. At this point, we are satisfied with just mentioning the one-carbon group transfers, but we will use half of this slide on glutamine amido transferases. These enzymes constitute a whole class of transferases that are not to be confused with the hepta or hexameric glutamine synthases in part 3. It may be useful also to remember the distinction between an amine and an amide. The amide is an amino group directly linked to a carboxylate carbon. You see it here. Now, glutamine amido transferases have two domains. One that binds glutamine and one that binds the amino group acceptor. The amino group is transferred through a specialized ammonia channel that keeps the reactive and potentially toxic ammonia from converting to ammonia and escaping. The end result of the reaction is glutamate, amines with one or two carbon chains attached, and either an alcohol or water. The outcome depends on the activated substrate. There are 20 amino acids, and all are derived from intermediates in glycolysis, the citric acid cycle, or the pentose phosphate pathway. I have indicated them with color here, here, and here. And they correspond to positions in this flowchart here, here, and here. Nitrogen enters these pathways through glutamate and glutamine, as we have spent some time detailing. Although the processes involved are similar from organism to organism, there is a significant variation in which amino acids a given organism can synthesize itself. Most bacteria and plants can synthesize all 20 amino acids, while mammals can synthesize only half. So, the non-essential amino acids are not needed in the diet. We can make them ourselves. But the essential ones, they are needed in the diet. So these are also marked with stars here. So you can read the table, or you can look at them in this flowchart and see where they come from. Some amino acids are also conditionally essential, and this means that they are required in growing animals or during illness. Synthesis pathways vary from simple to more complex. Generally, non-essential amino acids have simple pathways. We have already encountered some of these pathways before. Pyruvate from glycolysis leads to alanine in one simple transamination step. Another pathway we have encountered is based on alpha ketoglutarate that leads to glutamate and glutamine. We will follow it further to proline and arginine in the slides that follow. Ketoglutarate gives rise to glutamate and glutamine. You see it here. And this we have covered earlier. We are already familiar with ways that ketoglutarate can arise, most significantly as a product of the citric acid cycle. Also, the difference between glutamate and glutamine is just a transfer of one amino group. The enzymes involved have been covered earlier. So this leaves the pathway to proline and arginine, whose synthetic pathway branch at glutamate. You see it here. So glutamate can go towards proline or towards arginine. Proline is a cyclic amino acid where the R group turns back onto the backbone. It seems very unusual among the other 20 amino acids, but is actually closely related to glutamate if one considers its synthetic pathway. Its synthesis requires only 4 steps, 3 enzymatic and 1 non-enzymatic. In the first step, glutamate kinase adds a phosphate onto the carboxylate group, making it chemically susceptible for the next step. In the next step, gamma-glutamyl phosphate reductase uses NADPH to remove the oxygen and the phosphate attached to it, producing the aldehyde version of glutamate, shown here. This aldehyde is very reactive towards amines at acidic conditions, and there happens to be an amine very close. And that amine is actually on the same molecule, so this end can flip around and react with this, forming a ring structure. Now that the ring structure is formed, as P5C here, this process could in fact go backwards as well. However, another enzyme, pyrroline carboxylate reductase, is ready to seal the deal. In the final step, which is catalyzed by this enzyme, the final proline is produced. So one observation regarding this transformation is that while glutamate and proline is widely different with respect to chemical properties, nature easily converts it by supporting each simple step with enzymes and small biomolecules. Okay, finishing up proline and moving on to the biosynthesis of arginine from glutamate. Now, I will not go into the same level of detail as I did in the previous slide. That is, I will not rattle off all the enzymes and intermediates in the process. Instead, I will try to highlight some similarities. So the reaction on the top here constitutes the branch leading to arginine from alpha ketoglutarate and the citric acid cycle. What happens first is very similar to the first step in the proline pathway, except that we have an acetyl group added to the amine here. Now, in the first step in this part of the slide, a phosphate is added, exactly as in the proline case. It's added here at the end of the molecule. Now, different enzymes are involved here, working on different substrates, but essentially they do the same. Once the reactive semi-aldehyde is in place, it can be replaced in a transamination reaction here. The last step on this slide removes the acetyl group and produce ornithine. Now, ornithine is part of the urea cycle, which we will return to in a separate part. For now, we'll take a quick look at the urea cycle as it applies to arginine. Now, to the left here, in the gray background, you see the last product, ornithine, here, from the process on the previous slide, entering the urea cycle. On the right, you see a schematic representation of the urea cycle, and you can see that it's partly inside the mitochondria and partly in the cytosol. Now, ornithine is here. So I've indicated 4 steps in the process with capital letters encased in blue spheres. So ornithine is here in the leftmost part and here in the rightmost representation of the urea cycle. So by way of specific enzymes, you move from A to B, so from ornithine to citrulline, and then citrulline goes to arginosuccinate here. And then it sheds fumarate before it is converted to arginine. Now, this process, as we see, depends on the action of specific enzymes and on carboxyl phosphate, ATP, and aspartate. We can also note that excess arginine is one enzymatic step only from being converted to urea and excreted in the urine. That will happen here. So essentially, these two chemical groups end up in urea after the action of arginase. What remains after this is ornithine again. So we're actually back at point A, and this completes the cycle. In this lecture, we have looked at the biosynthesis of glycine, alanine, glutamate, glutamine, proline, and arginine. We could, of course, spend time going through all amino acids and all biomolecules containing nitrogen and their pathways for synthesis and degradation. I think that would not be a very productive way of spending lecture time. Instead, I encourage you to explore the book and solve problems related to the content of this lecture. And I encourage you to look for common themes in the reaction pathways and where metabolic cycles and pathways feed into each other and take products from each other. Now, I will round off amino acid synthesis by showing a slide outlining the synthetic pathways in bacteria that produce 5 of our essential amino acids that we need in our diet. I'm not suggesting that you should eat bacteria. It's just presenting it as an example of more pathways. And we'll do a small exercise where we look for familiar things. So I think that we already can see many familiar molecules that are involved in these processes too. So for instance, glutamate is turned into alpha ketoglutarate. That's a common theme, which by now we're familiar with. And in the final product, this glutamate through a transamination reaction has given rise to amine groups in green. So you see that the glutamate here is colored green, and the functional group that it gives rise to or donates is also in green. So pyruvate here donates part of its carbon skeletons to isoleucine, valine, and leucine. And acetyl coenzyme A donates another part of its carbon skeleton for making leucine through a branch in the valine pathway. So even though these pathways seems complex, and there are many of them, there are common themes that turn up no matter what organism you look at, no matter what pathway you look at. And I hope that can help you gain a handle on what is going on. Okay, so we've spent a little bit of time on amino acid synthesis. Now we will turn to amino acid catabolism. Amino acid catabolism is the breakdown to smaller parts and the subsequent processing of these parts. This is a process that is similar in most organisms. Now, just as amino acids could be synthesized from components derived from glycolysis, the citric acid cycle, and the pentose phosphate pathway, they can enter two pathways in catabolic processing: the glucogenic pathway and the ketogenic pathway. The glucogenic pathway slot into the citric acid cycle, giving rise to glucose. This cycle is also connected to the urea cycle through the shunt, which can get rid of excess ammonia in the form of urea. So if you have a lot of amino acids, you can actually put them into the citric acid cycle, shifting out the nitrogen and putting it into the urea cycle so that you get rid of it. Now that was the glucogenic pathway. Then there is the ketogenic pathway. So the ketogenic pathway feed into ketone bodies here. These ketone bodies are alternative energy sources that are made when glucose is in short supply. These can also be made from the breakdown of fats. Ketones are formed when there is not enough sugar or glucose to supply the body fuels. For instance, this could occur overnight. If you go to bed hungry and you sleep long, then you will not have glucose to fuel your body. Or it can occur during dieting or fasting. So amino acids lose their amino groups in this process to form alpha keto acids, and that is just a simple carbon skeleton, and this can be used by the body for fuel as well. Now, if your brain is completely full, you can take a break and return for the next installment.